To part 2 of our photosynthesis vodcasts. We're going to spend this one looking at the basics of the two different types of reactions that occur in photosynthesis, and we'll spend a lot of time in class talking about these. So here's where we're going to kind of enter at the ground floor and work our way through some of the more complicated aspects of this process. So here we go. Photosynthesis is broken down into two parts. We have our light-dependent reactions, and then we have the light-independent. We're going to focus first here on the light-dependent reactions because this is where it really kicks off. And so over here you guys can see that there is two different parts. So we have, there's where we have the light-dependent side. The light-independent side is down here, and we're going to get to that in a few. What happens in the light-independent reactions is that sun comes in, and the sunlight is there to basically drive a reaction to build up ATP. So even though we're building up sugars in photosynthesis, ultimately, we are actually creating some ATP molecules as a result as well. So this is an actual energy producing process. So in essence, that's what's happening. Sunlight energy is being converted into the form of ATP, and ultimately that ATP is going to help drive the production of sugar down the road. Okay, so the sunlight's real job is to really split water, and that's what we call photolysis. Photolysis, you guys already know. So sunlight comes in, and it's going to drive this fracturing of the water here. And so here's the sunlight coming in, here's my water, and here basically what happens is we break it down. Here's my O of my H2O, and here are my two hydrogen ions. Okay, so we get hydrogen ions, we get a single oxygen. The electrons then head into the proteins that are found on the membrane of the thylakoid, start bouncing through an electron transport system. And as they're doing that, okay, they are going to help build ultimately what we want, which is a lovely gradient of hydrogen ions inside here. We want a high hydrogen ion concentration over here. And they're also going to ultimately drive the formation here of one of our cofactors, NADPH. It's going to reduce it. Okay, so go back and think about, remember when we talked about oxidation reduction reactions, we're going to reduce NAD plus by adding in, gaining an electron, along with a hydrogen ion, and we're going to create NADPH, okay? And this is going to be very, very necessary in the light-independent reactions. We're also going to get the creation of an ATP molecule. Okay, so hydrogen ions are necessary to run through ATP synthase here, which is my enzyme. As it runs through, we get the connection of the phosphate to the ADP, and we create my ATP. So when we get that gradient, it's going to run right through. Okay, so this process is both cyclic and non-cyclic. The non-cyclic piece being that breakdown of the water, the cyclic piece is going to be the creation of our ATP and our NADPH. Okay, so just in the basics, that's that part. And like I said, we're going to do some more with it. So if you ever need to, you can always come back to this lovely diagram. It's also in your textbook as well. Now, the light-independent reactions become a little bit more complicated because this is a full-on cyclic reaction, okay? So what happens is, remembering, there's two parts of my overall reaction of photosynthesis. We just dealt with the water, right? So we've dealt with the water, okay? And notice here, if you remember, go back, we spit out oxygen, right? Isn't that one of our products of photosynthesis is oxygen? So when I split my water molecules and I release those hydrogen molecule that's released, poof, that's out. That's what the plants let go. So that's one of the products has already been established already in the light-dependent reactions. Where we're going to get the formation of the glucose, that other product is going to happen here in the light-independent reactions. So water's already in. Okay, that's my other part. The other reactant is carbon dioxide. So here's where we're going to see the carbon dioxide part of this. Okay, the ATP that we had is going to drive this energy process. It is a synthesis reaction, so it requires energy to occur. So the ATP is donating the energy. all right, for this, ultimately for what's going to happen. My NADPH is going to donate the hydrogen from this. So even though I reduced it in the light dependent reactions, now I'm going to oxidize it here in the Calvin-Benson cycle. My carbon dioxide is going to be there to donate the carbons and the oxygen that's necessary to help to create my carbohydrate. Okay, so here's where I'm going to have my hydrogen, here's my carbons and my oxygens to ultimately create my C6H12O6, my glucose. So where is this occurring? And this is occurring inside the area, or inside the space, inside the chloroplast, inside those thylakoids. So let's talk a little bit about, real quick, what's going on here. This is a great diagram because it really showcases where the carbons go. That's one of my biggest suggestions. Follow your carbons. If you can keep track of where the carbons are going, then you're going to have a pretty decent understanding. Because if you remember, conservation of matter, the amount of matter that goes in is the same amount of matter that has to come out. So I'm going to begin with 6 carbon dioxide molecules. Now, carbon dioxide is a single carbon molecule. It has 1 carbon. So 6 of those are entering into the cycle. It meets up with 6 of these rubisco molecules, okay? And that's what we call it, rubisco. It's got a much longer name, but notice a rubisco molecule is a 5 carbon molecule. So I'm going to take this 1 carbon molecule, we're going to add them together to create 6 of these 6 carbon unstable intermediates. And they're very unstable, they're not held together very well. Immediately, because of that instability, okay, they are going to break apart. So I'm not going to have 6 carbon molecules, I'm now going to have 12 3 carbon molecules, okay? And these are known as PGA, okay? This molecule heads down as it's part through the cycle. What's going to happen, okay, is it's going to be converted using energy, okay, where we're going to use up 12 ATP molecules, okay, that are going to donate the energy for this, 12 NADPHs, okay, so that is going to help reduce and donate C6H12, okay, just throwing that out there, 12 of these, okay, is going to take this hydrogen, okay, we're going to attach those on. We've gone from being PGA now to 12 P-gals, okay, and then these are going to swing right around. I'm going to take 2 of these, break them away, okay, so where I've gone from 12, now I have 2. These 2 are going to get basically combined in a way to where I go from a 3 carbon to 3 carbon molecules to 1 6 carbon glucose that's been phosphorylated. All right, so I know you guys probably, your eyes are going crossed right now. Again, we're going to work with this, okay? Now, all of these 2 reactions here, and you can always go back to relook at them again, but we keep talking about sunlight, and sunlight has a whole lot going on with it. There's tons of different types of radiation that occurs in light, and our visible light is what plants use. And all our photosynthetic organisms use the visible light spectrum. And it's a very, very small chunk of the overall light that we have. And if you've ever taken light and passed it through a prism, that's where you can see those visible light wavelengths. You can see all those colors of the rainbow. So only about 1% or so of it gets used, which is pretty amazing given the fact that light is so abundant. That's why we have so much energy that hits there. And you have your electromagnetic spectrum, you have gamma rays, which are your radio waves. We have, in terms of the visible light, what we care about is the range that goes from about 400 to 750 nanometers, and that's going to be what we see. That's what our eyes are capable of seeing and what the plant is actually capable of using, which is pretty nice. So light's really cool because it has 2 types of properties to it. It has particle properties and it has wave-like properties as well. That's what makes light really, really neat to study. And if you get a chance to go back and look, part of our understanding of light and the photons that create light was what won the Nobel Prize this past year in physics. So those wave-like properties are pretty impressive. It also has those particle-like, and that's where we get the photons. And that's what won the Nobel Prize, is the study of photons. And photons are used to, basically, those are the particles that the plant uses in photosynthesis. So the higher the wavelength of light, like in your reds, your 700s, your 750, you get low energy photons. High energy photons are the other way around. Okay, so those are going to be your blues. Plants like reds and blues primarily. At least I should say chlorophylls like the reds and blues primarily. They do not like to absorb green light. Okay, so what I want to show you is an experiment that was done by Engelmann that helped us understand what particular wavelengths of light plants really liked to use. Okay, so here we have a prism. What he did is he took a prism, split the visible light spectrum down. He placed on it algae. Okay, so here's photosynthetic algae. That's these right here. Here's one cell that you can see. Here's another cell. Okay, so you can get the gist of it. Those are the algal cells stretched across a prism. Okay, and so what he did there, so this drop, he used the water to do this and everything, places it on a slide, puts the colors out there, and then allowed bacteria to kind of be spread around these algal cells. What he found was that the most population of bacteria occurred in this area, occurred over here in this area, and down in this area. So why might that be? Why were we seeing more bacteria down there with very few in the center here and in these areas? Okay. Reason being was these algal cells were using the blues and the reds much more effectively, producing far more amounts of sugars as a result. That fed the bacteria. The bacteria grew in greater population. So the more bacteria you saw, the more available energy source, the more available sugar. Fewer bacteria, not so much. So from this, we learned that plants, especially the chlorophylls, are more capable of pulling in and absorbing those blues and reds in terms of the spectrum. So pretty cool. All right, so as a result of this, we learned about some of those plant pigments and the fact that they absorb very specific light wavelengths. Our chlorophylls, which tend to be the ones we see the most, as I mentioned, are your blues and your reds. And as a result of absorbing, that means they're going to reflect back the other ones. So we mentioned this in class. Why do we see leaves as green? Why does it appear green? They're not absorbing green. Instead, those green wavelengths are being bounced back off, and that's what's entering our eye, and that's how we see them. Okay, so in terms of the spectrum of the pigment, we can see here, here's a number of different types of pigments with specifically your chlorophylls, because those are the most important. Notice their most absorption is happening in here and in here, okay? But yet there are other plant pigments too. There are keratins, so your beta carotene, okay? Those happen to really like blues. They don't like the reds so much, so they're gonna bounce back oranges, hence why a carrot appears orange. And there are a few others that are out there as well. But so here's our chlorophyll, and chlorophyll A and chlorophyll B are two chlorophylls that are going to be very important pigments, which means are proteins, for the absorption through the different photosystems of the light-dependent reactions. That's where it's the most involved. It sets up those pigments that are going to, that electron transport system that we mentioned earlier. Beta carotene, a little bit different, okay, in its structure. So and we have our chlorophylls, which are right there. Some other pigments are your carotenoids. Like I mentioned, those are the ones that are involved in absorbing mostly blues, your blues and your purples. Xanthophylls, these ones are pretty interesting. These are your yellow pigments. And so that means that they're really liking those. They're a derivative of the carotenoid group, and when you actually do chromatography of plant pigments, you actually can see a yellow band because they really like to absorb the reds primarily. Then you have your phycobilins and your anthocyanins. These are pigments that are primarily found in red algae. They capture reds and yellows in terms of light. Your anthocyanins which I just love the word. Anthocyanin means blue flower. They appear purple. These are some of the pigments that are going to show up in like flower color, they're going to drive for like insect attraction. Anthocyanins are also known to be acting as a sunscreen for a plant because they'll absorb the blue-green light, and they also absorb ultraviolet light. So they kind of act as a light protector for the plants, which is pretty neat stuff. That's just your overview. We're going to spend time in class with the actual reactions. So go back, check it out if you need to, okay? And I'll be explaining a few more things in class. But as an overview, there you have it. Have a good night, and we will have one more on photosynthesis later on. So take it easy, and we'll catch you later.